And this will look at what kind of genetic and genomic testing is available for patients, what samples are taken, what results actually mean when you get some from this, and then try to summarize it in a way that lets us be aware of both the risks and benefits of genetic testing. So let's get started. Defines genetic testing as the analysis of DNA, RNA, and chromosomes, proteins, and even protein metabolites to identify heritable variations in genes and/or chromosomes. Why do we do genetic testing? For we can, at this point, clinically diagnose more than 2,000 disorders. We do it for research purposes, and now recently we're doing it for recreation, using direct-to-consumer genetic testing, mostly for ancestry, but also for some genetic disorders as well, which of course gives us professionals a little bit of pause and anxiety, to say the least. Positive results in a genetic test, in a clinical test. What does that mean? Well, if you have a patient that has the deletion F508 in the cystic fibrosis transfer regulator gene, then yeah, they're going to probably develop cystic fibrosis. But a lot of times they will have a mutation or polymorphism that's simply associated with the disease, in which case they may be only at a higher risk. So where are you going to get information, reliable information on these tests? Well, there's a website that is funded by the National Institutes of Health that has a fairly up-to-date list on this, and I would recommend going to that. And you just have to click on www.genetest.org, and when you really want information, that's where you should go. Samples are used for genetic testing. Well, the most common is a buccal swab, getting cheek epithelial cells, but they can also take just a blood sample. And of course, what they want are the white blood cells, which have a nucleus. The red blood cells do not. The different types of genetic tests are going to really tell someone something different. For example, if you're going for diagnostic testing, diagnostic testing is going to confirm or rule out a diagnosis in a person with symptoms. Okay, so they have clinical features, say, that are suggestive of Marfan. They're going to do the genetic test to confirm it. Diagnostic testing can also be based on standard or clinical or biochemical tests. For example, for cystic fibrosis, they do the sweat chloride test first, and then if that's positive, they may follow up with genetic testing. Anyway, the most important thing to remember, though, is that genetic testing, particularly diagnostic, is going to give information about the patient that has symptoms, but also about the other family members as well. So you have to take that into account. Now, what about predictive testing? These are for patients that are asymptomatic, but am I going to get this in the future? So what happens if a pre-symptomatic test is positive? They, in general, if it's a predictive testing, they will get the disease. The classic example of this is Huntington disease, which is a very nasty disease with age-related penetrance. It usually doesn't occur until the 40s or 50s, maybe a little bit earlier. And of course, it has some serious implications for length of life, etc. And then predispositional testing, and this unfortunately is what a lot of these direct-to-consumer tests are. They're looking for gene variants that seem to increase our likelihood that we'll get a disease. But people will not necessarily understand that it's just increasing them. But this is true for mutations in BRCA1 and 2 for breast cancer. The risk is 85%, which is fairly high, but it isn't 100%, and that's important to remember. Who's had diagnostic, predictive, or predispositional testing is faced with getting an inconclusive result. What does that mean? Well, it means they have a gene variant, but it's not linked to disease. The variant is located within the same gene as other mutations that induce the disease, but it's different from the mutations that are linked to the disease and different from mutations that are not linked to the disease. So it really is inconclusive. What should that patient do? Well, they need to continue high-risk monitoring, particularly if they're looking at something like testing for cancer, predispositional testing for cancer. And they need to receive counseling because they're very likely to be frustrated and confused, if not a little angry as well. Well, getting back to the different types of testing, you can also have what's called carrier testing. Obviously, these are people who have affected family members. They don't have the disease themselves, but typically they're worried about starting a family. And this is particularly true for certain ethnic groups that are at higher risk for a cluster, usually, of certain genetic disorders. A perfect example of this are if both parents are of Ashkenazi Jewish descent, basically Eastern European, they do what's called an Ashkenazi panel. This panel is really looking for disorders that are either autosomal or X-linked recessive, and you want to test both parents to see if they are both carriers. And of course, again, predominantly carrier testing is done for family planning purposes. Now, within the same genetic disorder, mutations that lead to disease may differ among different ethnic groups. For example, the cystic fibrosis gene is of itself a very, very large gene, and there are many, many different mutations that are located in there. Some, of course, with severe disease, some with mild disease, some with no disease. Okay, so it's frequently, if you have a particular ethnic group with the parents, you will look actually for different mutations in the different ethnic groups. The slide just shows you a list of some of the different disorders that are done. Here is the Ashkenazi Jewish panel. You've got Bloom, Cynefin, cystic fibrosis, Gaucher, mucolipidosis, Niemann-Pick, Tay-Sachs. And you can see here some even without the Jewish, the Tay-Sachs and the thalassemias, etc. So you're looking for different diseases in different ethnic groups, and you look for different mutations of the same disease in different ethnic groups. Testing, of course, is prenatal testing. These are usually requested from pregnant women. They're looking for specific gene variants, or if they're older, they're looking for the presence of chromosomal abnormalities. During the first trimester, which can include 9 to 13 weeks, mostly what's done is blood test from the mother. They'll look for maternal serum alpha-fetoprotein levels. If they're abnormally high, this might be suggestive of neural tube defects. And then they do another with alpha-fetoprotein plus unconjugated estriol and HCG. Those can implicate Down syndrome, in which case you do some more. But these are all screening tests. If something seems somewhat abnormal, you would go on to do additional testing in the second trimester. And now you can actually start to look at the developing child, the ultrasound, echocardiography, MRI, fetoscopy, and of course you're looking for general features. When you get to 10 to 12 weeks, you can do some tissue sampling through chorionic villus sampling, amniocentesis after 4 weeks. Both of these, of course, have a small risk for abortion and infection, but it's usually mild. And of course, again, it's a situation where the mother's usually older and they're concerned about, or there's a family history of certain disorders. And so they want to know whether they have a child at risk, or whether how they need to prepare for the birth of a child with multiple or serious problems. Shows you some of the different techniques where the sampling is done with prenatal. You can see an ultrasound versus going for amniocentesis versus chorionic villus sampling. Testing is somewhat controversial. It's called pre-implantation genetic testing, or PGD. It's done along with parents who are undergoing in vitro fertilization. They take egg and sperm from the parents and they, incubate them. And then with the fertilized eggs, as they start to develop into, when they get into, I should say, the 8-cell blastocyst stage, they remove 1 or 2 cells and then test it for single gene and chromosome disorders and determine also the sex of the child. Then based on that, they'll choose the healthy embryos for implantation, usually up to 1 to 2. If it's X-linked recessive in the family disorder, they may only want females. This, of course, is usually done with older parents, and it's very expensive. But, of course, the problem with this is what's going to happen with the unwanted embryos? Do you just dispose of them? And this, of course, is where a lot of the controversy occurs, and whether this is really something that should be done, and not even just only want females, because there's an X-linked recessive. There's a lot of difficulty with some of these procedures for many people. Screening, another type of genetic testing. Of course, this seeks to identify infants at high risk for disorders, particularly some of the metabolic disorders we learned about. And these are then kids that would need treatment shortly after birth. The tests are typically biochemical as opposed to genetic, looking for high concentrations of metabolites, showing that they are deficient in a particular enzyme. It was begun in the 1960s by Dr. Robert Guthrie, who developed the test for phenylketonuria, PKU. These newborn screening tests are supposed to actually protect the public health and are going to differ in each state because the states pay for it. But depending on the state, they can screen up to 30 disorders, and this is becoming a lot easier to do and faster to do because of a newer technique called tandem mass spectrometry. We'll talk a little bit about that in a second. Anyway, all of the disorders that are commonly tested for are autosomal recessive. So we're talking about PKU, it's sickle cell anemia, galactosemia, which we didn't really talk about so much in our inborn ears, but I did mention it. And then, of course, congenital hypothyroidism. Whether we should screen for a given disorder or not. What are the criteria? First of all, is this a disorder that produces irreversible damage if we fail to untreat it, or not treat it, I should say? The treatment prevents damage, but only if it's begun very early in the newborn stage. The natural history of the disease must be understood and known. A screening test, of course, must be available for it. And the facilities for diagnosis and treatment are available. So some of the newer techniques allow analysis even in a single sample, and that's what we'll see on this next slide. So tandem mass spectrometry basically is you metabolites that are present in the blood serum are broken into these little fragments, and then they're kind of their mass is determined, which will also determine in part their chemical formula stuff. And so you could simultaneously look at a number of different metabolites very, very rapidly. Some of these are fairly well standardized, some of them are still being worked on, but it is a really marvelous technique where we can get the answers a lot faster than we used to. And so I think you'll be seeing this being used and referred to more and more in days to come. Types of genetic testing include zygosity testing. Are we looking at monozygotic versus dizygotic? So are we looking at the recurrence rates, concordance rates, parentage testing, determining the biological parents. Will also help finding out someone's medical history if they're adopted. Now, one of the things you should be saying to yourself, who's making sure all this genetic testing is being done properly? Well, first of all, the major one is the Clinical Laboratory Improvement Act, or CLIA, which certifies the clinical labs, will certify clinical labs, I should say, but not research labs. Then you have the CDC, the FDA, Centers for Medicare and Medicaid Services, and the Office for Human Research Protection. So this alphabet soup also makes sure that genetic testing is reliable and reproducible. And then the United States Department of Health and Human Services, which of course is in charge of the NIH and things like that, are also working to improve oversight of genetic testing. And especially when it comes to direct-to-consumer, where there's very little oversight at all, this is something that should be a real concern to medical professionals, because people are letting their DNA be sequenced and tested, and they don't know where it's going, and they don't know how to interpret it, nor do the people who are doing the testing, nor how good the results that they're getting is going to be. So there are a lot of real serious concerns with this whole area of medicine. Let's put those concerns aside and find out what some of these tests are. First of all, DNA sequencing. We're just figuring out the actual linear sequence of bases in the entire genome, or specific genes on specific chromosomes or stretches of chromosomes, anything that carries a lot of variations between individuals. That's one of the things they frequently focus on. In order to do this, they need cells with a nucleus, because if you have cells without a nucleus, then you have a cell that doesn't have any DNA, and you're trying to sequence the DNA. So yeah, you gotta have a cell with a nucleus. Anyway, as I've said earlier, white blood cells and buccal epithelial cells are our two major sources. Now, there's not going to be a lot of DNA in those samples. So what do? To increase the amount of DNA present when you don't have a lot, well, you have a little test which made the man who developed it a millionaire, called the polymerase chain reaction, or PCR. It takes small samples of DNA and replicates them, so you get tons of it so that you can sequence it and study. And that's what's always being used on those things like CSI, those TV shows like CSI and everything. These are two of the common techniques that they're using. And I couldn't resist putting in a slide that, of course, this is all automated now. All of these different colored peaks represent, T, G, C, and A. This are A, G, C, T, and the colors. And then the computers, of course, read this out and put it in sequence. And this is how we're able to sequence the entire human genome. Now we can do what took us 15 years the first time. Okay. It now takes us somewhere a couple of hours to do. And pretty soon they estimate that it will take us maybe about 30 minutes to sequence an individual's entire genome. 3 billion base pairs. That's how automated and sophisticated this has become in a very short period of time as well. Already talked about cytogenetic testing, karyotyping, where we're looking at the chromosomes themselves with a metaphase spread, and of course we're looking for chromosomal abnormalities. Another one we mentioned but didn't really talk about is called fluorescent in situ hybridization. We call it FISH for short, of course. It's really kind of neat because we've got these complementary probes. Remember that DNA is complementary, so if you have A on one strand, you have T on the other, and they will just spontaneously form hydrogen bonds and stick together. So if you label a probe that's complementary to a sequence, okay, in individual chromosomes or something, you can make them fluoresce different colors and identify the chromosomes and potential abnormalities as well. So you can see, again, like the karyotyping, abnormalities in chromosome structure and number. And we see an example of this on the next slide. So as you see here, this is what we call FISH. Sometimes it's referred to as chromosome painting. On this side, it's a little more sophisticated. And we see here a normal male. On this side, we see an abnormal female. And if you take a look, what you're seeing is a couple of unbalanced translocations. Actually, I think just one. I'll take a good look. Oh, there's more than one. Yes. There's a couple of things here that are a little weird with this abnormal female. See if you can find it. We talk about genome-wide association studies, or GWAS. So what we're trying to do, like we were talking about with the psychiatric disorders, find areas of the genome that are associated with the specific disease. Okay, we're going to use various genetic markers so that we know that's present on the different chromosomes. So these can be genes, but a lot of times they're single nucleotide polymorphisms. In other words, it's just a mutation. The normal sequence is AGCT, but if now we have AACT, okay, that's a single nucleotide polymorphism, abbreviated SNP and referred to as, yes indeed, SNPs. Okay, really, they really do call them SNPs. Now, the computers will compare the sequences of people without the disease to those with, and we're not talking just about 2 or 3. We're talking about thousands of them. So all of this was not. We weren't able to do this until the computers were powerful enough to do this kind of comparison. And so we look for regions, and we look then at the genes in those regions. And this is what's giving us biologically plausible genes to then investigate to see if they really do contribute, say, to a cardiovascular disease or hypertension or schizophrenia. This is how they're doing it. It's just showing you how we screen for SNPs. You would have patients over here on your left, and they would get the DNA and chop them up, and then have them stick to various gene chips that have the genes on there, and they would see which ones would bound, and then they would fluoresce different colors, and then you compare them to those that don't have the disease and look for differences and see which are associated and which are not. Okay. Again, because this is computer generated, the information, it's a lot faster than what we would have to do. But we have a lot of data right now, which some of it is, fairly well interpreted. Some has a lot to go before we get there. Which leads us to a final topic, really, of talking about these direct-to-consumer genetic testing. And needless to say, the American College of Medical Genetics and the American Society of Human Genetics are very unhappy with these companies because they really believe that you need a professional to interpret the results, and they are not truly giving the patients informed consent, and in many cases just simply doing inappropriate testing. So also complaints that they have are that these DTC genetic testing companies lack follow-up care. They're misleading in what they can and cannot diagnose. And another thing, are they really protecting their consumers' privacy? I know some people want to have less government, but there are times when government has a pretty good role, and this can be oversight watching. There's no federal oversight for this right now. And I think in the years to come, we're going to think we really need some. And it's expensive. Why should people throw $1,000 away on something that's just going to make them upset and they don't really know how to interpret it? So the most important thing is, if you've got have a patient who's concerned about a genetic disorder, guide them to a professional, not to a direct consumer genetic testing company. What are the risks and benefits of genetic testing? Well, let's say you have a family that has an increased risk for colon cancer. So you're tested, you don't have the family variant. Well, that doesn't mean you never have to be tested for colon cancer. It just means that you can be screened at the same rate as the general population. Okay? I always have them talk to a counselor before getting any kind of genetic test, so that they know what a positive, negative, and inconclusive result means. Make sure they're going to a professional, a genetic counselor or nurse or medical geneticist. They should be told their options if the test is positive. It doesn't mean they're going to die tomorrow necessarily. Discuss the different types of genetic tests and what they can and really cannot tell you. Make sure they're getting their genetic testing at a major medical center or a private clinic Usually it's organized by specialty, at which point if they need to consult a directory, have them go to www.genetest.org again, because that's the place to find the most up-to-date and reliable information. And then make sure that they are aware of their privacy, the importance of informed consent, and confidentiality if they agree to this testing. So we've covered a lot really in this chapter of just about the different types of techniques, how they're done in the laboratory, and how they can be misused or misunderstood by the companies and the consumer. So that's it for this particular week. Next week is our last week devoted to genetics, where we'll be looking more at some of the ethical considerations that are involved in genetic testing. And then we move on after that into immunology and microbiology. So until next week, this is Dr. McDonald. Goodbye.